Hey guys, it's Kavya and welcome to my YouTube channel. Did you know that you can make milk using yeast? Yeah, you heard me, yeast. In today's video, I'm going to talk about the process that companies are using to create this animal-free milk, and I'm going to demonstrate a quick tutorial on how to use Benchling with the Digest and Ligate method. It all starts with cellular agriculture. Cellular agriculture is the technology behind creating products without animals. Well, mostly without animals. There are a few exceptions, like the fetal bovine serum, but that's a discussion for another day. Milk is made out of two main proteins, one being whey and the other one being casein. It is important to note that there are many different types of these proteins, like beta casein and kappa casein, and there are different types of whey as well. You've actually probably heard of whey before. It's like whey protein shakes, the stuff that you drink before workouts. So anyways, these cows carry the genes that encode for whey and casein. Since the scientists already know about this, we don't really need the cow anymore. One of the genes that encodes for casein is called the CSN2 gene, but there are several different types. The process goes like this. First, they get a yeast plasmid, which is essentially just a circular piece of extra chromosomal DNA. They take out a piece of that plasmid and insert the cow gene into it that encodes for the whey and casein protein. Then they insert it into the yeast cells, and these yeast cells start to replicate and grow. As they replicate and grow, they're going to produce whey and casein. After scientists have these whey and casein protein molecules, they can mix it with other substances. And there you have it, animal-free milk. Pretty cool, right? Here's a visual for my visual learners. The process of acellular production would not be possible without fermentation tanks. Simply put, fermentation tanks are really big tanks, like the ones found in breweries, and their purpose is to help the cells grow. This is really achieved by setting the tanks at the proper temperature, pH levels, and the salinity. And then, when they're ready to be taken out, the cells are mixed with different types of sugars and other substances which eventually lead to milk. Fermentation tanks are the key contributing factor to the scalability of this industry. They're acting as if they are the cow, which is enabling these cells to produce these mature proteins. So, in order for you to see animal-free milk and other animal-free products in your local grocery store, the supply of these fermentation tanks needs to increase, while the cost of actually purchasing one of these needs to significantly decrease. Remember, the technology of fermentation tanks has been around for almost 40 years, but the way that this industry is actually using this technology is truly astounding. Alright, since I've gone over how people are creating animal-free milk, let's move on to the Benchling tutorial, where I'm going to do a quick demonstration on how to use the Digest and Ligate method.
Alright, so now we're going to start setting up our plasmid by inserting a new DNA sequence, and you can just name it the name of the chosen plasmid. And once you've named it, you can go ahead and click Create, and here's the important part. You want to copy and paste all your base pairs, and Control-V should help you do that. And then, so now that we have our plasmid, we can also insert our gene. So we're going to do the same process, but this time name your entry name the name of your gene. So in this case, it's actually a fake gene. And then you can go ahead and click Create. And we're going to do the same thing, just Control-V. And now here's the fun part. We're going to start a Digest and Ligate assembly. And we're going to make sure that you're on the backbone and click on the two restriction enzymes that are highlighted. And then you want to invert the selection and set it from the selection. And you can see that the base pairs are lining up. Then you want to go into your insert, and then you want to click on both of your restriction enzymes by clicking Shift and then holding down. And you want to set it from your selection. And once you see here everything's lining up, so you can press Done, and then you can name your new plasmid with your insert in it. and you can just click the folder that you want it to go into. And there you have it, your new plasmid with your insert.
Thank you so much for watching my video. If you enjoyed it and learned something awesome about cellular agriculture, be sure to tell me in the comment section or like the video. Bye guys, see you soon.